Everyone to Monday afternoon plenary lecture. I'm more than delighted to introduce today's lecturer, Ivan de Araújo. Looking into his CV, he's a person of multidisciplinarity, I realize.
True.
So he studied philosophy and artificial intelligence and did his PhD in medical imaging and experimental psychology, so already many disciplines. After doing his postdoc, he moved on to head labs at Yale and at Mount Sinai Hospital, until he moved in 2023 to Tübingen, to the Max Planck Institute for Biological Cybernetics as a director. Yeah, I very much look forward to his talk. And before I start, please pay attention to the QR code that just disappeared, with which you can put the questions that we can then select at the end of the talk. The floor is yours.
Thank you, So-Young. Thank you so much for the invitation, John, and all the organizers. I'm really happy to be here. So, as you may all have heard from the news and scientific papers, our gut, our gastrointestinal tract, contains an incredibly large number of microbes of many species and types that live almost permanently in this organ. Some of them perform very important work for our health. For example, helping to digest food and increase gut motility and so on. There is one particular type of microbe that lives in our gut known as lactobacilli. They have this name because they produce lactic acid. But they are very important for certain physiological functions that we are now starting to understand. So this is a cartoon illustrating the organization of our gut. So here is the lining of the gut. If you imagine the food comes here, nutrients and vitamins are allowed in by these epithelial cells that cover the gut, that protect the gut against the external environment. And then things that we want to have in our bodies crosses through the epithelium and find themselves in the blood circulation. So this is basically absorption. And we want to keep out things, substances, and living bacteria that can be harmful to us. That's why the epithelium of the gut produces certain proteins called tight junction proteins that keep the epithelium tied together, not allowing for potentially harmful elements to cross into the blood. So in a healthy gut, we have a certain amount of mucus, a gel, glycan, that allows for bacteria to live in and proliferate. So lactobacilli, they need this mucus to survive in the gut. And for reasons that are not very well understood, lactobacilli produce metabolites that enhance the expression of tight junctions. So the presence of lactobacilli is very important for protecting the blood against elements in the gut that we do not want to have in our bodies. For reasons that are very interesting, potentially very interesting to neuroscientists, this whole system seems to collapse when psychosocial stress sets in. So lactobacilli numbers seem to decrease dramatically, and also the interaction between bacteria and mucus seem to decrease dramatically whenever certain types of stress set in. So early studies on maternal separation in monkeys, for example, were some of the first to show this phenomenon. Prenatal stressors in humans and animals have been shown to decrease lactobacilli numbers. Certain types of psychosocial stress in humans, for example, bullying in school and social isolation in children. So all these types of stress, they reduce lactobacilli number, then they lose the tightening of the gut, and they increase, very importantly, the susceptibility to infection in these individuals. And one other example of this situation that is being driven by stress, at least worsened by stress, and that causes an opening of the gut barrier, is inflammatory bowel disease. So there is some sort of connection between psychological states, in particular stress, and the ability of the gut to protect the blood, the internal organs, from invaders in the gastrointestinal tract. So in fact, if you look at a healthy animal, allow it to explore, say, an object like a tube, if we count the number of lactobacilli in their gut, we are going to see a relatively large amount of lactobacilli living in their guts, in male and female, so blue and pink here. However, if you expose animals to certain types of stress, for example, restraining them in a tube for 1 hour a day, then these numbers of bacteria decrease dramatically in both male and female. So there is a graded response, especially in males, to an acute stress, but in chronic stress, if you repeat the experiment every day for a couple of days, then you basically cannot count in culture any lactobacilli. So this is what had been shown before. To prove that this lack of lactobacilli affect the ability of the organism to protect itself from external events, you can see, so if we inoculate the animals with harmful bacteria, for example, Staphylococci, then we see that in healthy animals, we can barely detect this Staphylococci in the blood. So if we culture the blood for this harmful bacteria, we can't really find them in the blood, which is the normal situation. However, in the stressed animals, we see that a large number of these bacteria will be able to cross into the circulation, so that if we count their numbers in blood, now we see an important effect, an important amount of bacteria are present in the blood, causing systemic infection. So just to reiterate, these stress situations open the barriers of the gut and allow for harmful things such as bacteria to get in. Another evidence for the weakening of the immune system in these animals is that if we look at the size of the spleen is a lymphoid organ that stores immune cells for the event of the need of a fighting infection. So if we compare with the stressed animals, we see that the spleens have been contracted, meaning that the immune cells that were present in the spleen were recruited by tissues in the body to fight infection. So there is a very obvious relationship linking stress via lactobacilli to the weakening of the immune system. The question is, how does that work? What is the potential link between psychosocial stress, psychological states, and the weakening of peripheral immunity? So here enters the vagus nerve. So the vagus nerve is the largest of the cranial nerves in the body. Is a nerve that originates in the brainstem, and travels out of the skull towards a number of organs in the periphery, especially the gastrointestinal tract, but also the lung, the heart, and some other select organs, visceral organs. So many years ago, more than a century ago, it was discovered that stimulating the nerve could improve, could decrease the number of seizures in epileptic patients. So this became a standard treatment for medication-resistant epilepsy in several countries. Eventually, however, they noticed that the same treatment could improve mood states in major depression patients, especially those who are resistant to medication, and especially those in more serious cases with suicidal thoughts and so on. So also in many countries, I believe in many countries in Europe, in addition to the US and also in Asia, this treatment is approved for major depression patients that are not responding well to certain medications. So this is just a electrode implanted in a nerve that stimulates the nerve repeatedly. But there is another interesting aspect of the vagus nerve stimulation that may link to what I was presenting before, which is the fact that stimulating the nerve has an anti-inflammatory effect. So it has been shown in patients and animals that stimulating the nerve improves a number of immunological parameters in the circulation. In particular, it helps to decrease certain inflammatory markers that are present in these patients. So we hypothesized that one way to link psychological states to these alterations in gut mucosal immunity could be abnormalities in the vagus nerve activity. So, as I mentioned, the nerve starts in the brainstem and goes all the way down to the gastrointestinal tract. So here, this is a cartoon representing a section of the brainstem. And here in this little area called DMV, there are parasympathetic neurons, vagal neurons that are the main drivers of parasympathetic activity that innervate large parts of the gut. So the first question we asked was whether stimulating these parasympathetic neurons in the brainstem associated with the nerve would have any effect in the immunological parameters that were altered by exposure to stress. So we used the technique called chemogenetics, which has some technicalities, but the important point here is that here, if you look at the same section in the brainstem, these red neurons here, which now express a foreign receptor, they are the parasympathetic neurons that from where the vagus parasympathetic innervation originate. So using this technique, we can, at our will, stimulate very specifically these neurons and no other neurons in the brainstem, and look at the effects in vivo. So when we stimulate these neurons in this what we see is that a very striking increase in the number of lactobacilli present in the gut. So here is the stressed animals. There is barely any lactobacilli, but in the same stressed animals, if we pair stress with activation of the vagus nerve, we restore the normal numbers or normal counts of lactobacilli in the gut. In the same way, by stimulating the nerve, we are no longer able to detect harmful bacteria in the blood of stressed animals, meaning that stimulating these parasympathetic neurons was sufficient to increase or decrease the permeability of the gut epithelium. And finally, consistent with all that, we observed normalization of the size of the spleen, meaning that the spleen is no longer forced to release immune cells into the circulation to fight infections that may be going on in the body. So we have one element of this system, which is that the vagus nerve seems to be implied in the mucosal immunity abnormalities that are observed in stressed organisms. Next, we wanted to find out how exactly the vagus nerve could do that. And we performed some viral-mediated tracing studies looking at whether these vagal neurons, these parasympathetic neurons, can control mucosal glands that release the mucus that the bacteria need to survive in the gut. And we performed these studies, and just to explain roughly what we found is that there is a group of glands in the upper duodenum known as Brunner's glands that are very poorly studied, but these glands are very particular because they seem to be the only mucosal glands in the body, or at least in the gastrointestinal tract, that are directly innervated. They need neural inputs to release mucus in the gastrointestinal tract, unlike the other mucosal glands that live more on the surface of the gut and do not receive these types of inputs. So we started to study these glands more carefully, and you can see here an illustration. So they are concentrated here in the pyloric junction with the duodenum. If you look at this cartoon here, this is the epithelium, this is the surface of the gut. So the glands live in the submucosa just below the epithelium. You can see them here in this tissue picture, or maybe it's easier to see in this coronal plane, where this is the surface of the gut, and you can see the glands underneath. So the arrangement is such that the innervation reaches the glands here and they release mucus in the surface rather than releasing mucus in response to elements in the surface of the gut itself. This is how they look in humans. This is from a tissue from a pediatric patient, and they look very similar in humans compared to other species. And interestingly, these glands only exist in mammals. And in every mammal, the glands are in the same place and they have the same extent or same degree of innervation. So in every species, they seem to be under the control of the nervous system. We then developed some molecular techniques to eliminate these glands specifically from the stomach. There is a lot of elements to it, but basically we found that if we remove the glands from the intestine of animals and using molecular techniques that do not damage other cells, and we inoculate the animals with harmful bacteria, then basically all of them will die within 5 days of generalized infection. Basically exaggerating the effect of stress. So if we look for the presence of this bacteria in the blood of these animals, we see a very significant increase of numbers of bacteria in the blood of these animals, meaning that these glands, which is basically something that was not known, these glands are very important to protect the body against pathogenic invaders in the gut. So they basically exaggerate the effects of stress, and then we, of course, were very interested in the possibility that they are related to the immune compromise that takes place during chronic stress. So in order to study the communication between these glands and the nervous system, we developed a system that basically combines the ability to stimulate neurons and the nerves in the organism, and at the same time we can use calcium imaging, which is a technique to look at the activity of cells in various parts of the body, including the glands, the Brunner's glands in the intestine. So we could, in the same animal, perform recordings and stimulation experiments and observe the effect of these manipulations on the behavior of the glands in vivo. So for observing the activity of the glands, we implanted a glass window over the abdomen of a mouse. They are fine basically with this implant, and this opens a window into a living organism, into observing how internal organs, internal cells behave in vivo, which is something that we need to advance in science to understand better how the nervous system controls peripheral organs. So here is a movie showing the activity of the glands upon stimulation of the vagus nerve. So we basically implanted the same electrode that is implanted, the same type of electrode that is implanted in patients sustaining depression or epilepsy treatment, and we send pulses through the nerve in a similar way, and at the same time we used confocal microscopy to observe the behavior of the cells of the Brunner's glands in vivo to test the idea of whether the nerve is driving the cells. And you can see the signal is very potent whenever we stimulate the nerve, meaning that these cells are potently secreting mucus whenever the nerve is stimulating, explaining to this point the ability of vagal stimulation to enhance the number of lactobacilli in the gut, because the lactobacilli need the mucus that is being released from these glands. So we think that one potential pathway via which the vagus nerve may produce its effects on major depression patients is by breaking this cycle that usually starts with dysbiosis caused by stress, that then disrupts the immune system of patients and potentially influence mood and psychological states via alterations of the immune system. So one potential mechanism to explain why the vagal nerve stimulation changes the immune system, which is something that remains poorly understood, is by first affecting the state of the gut and then improving the overall immune outlook of the patients. Finally, we wanted to know what is the role of the brain in this system. So we continued with our tracing studies, and we looked for brain areas that could be controlling the glands via the vagus nerve. So we found one particular nucleus in the brain that seems to be anatomically very strongly connected to the vagus nerve and the Brunner's gland in particular, which is the central nucleus of the amygdala. So the viral expression in this part of the brain was very strong whenever we placed the viruses in the glands themselves in the intestine, and this effect disappears if we cut the vagus nerve. And we found this to be very interesting because the central amygdala, as many of you may know, is one of the brain areas critical for emotional processing. It has been implicated in fear, stress, and anxiety for many years, based on the study of many investigators over the years. So we thought that the central amygdala could be a very important node in the central nervous system that in which activity alterations produced by stress could impact on the activity of the glands and then eventually impacting on the immune system. So we went on to look at this possibility on the functionality of this circuit linking this emotional nucleus of the brain, the parasympathetic neurons of the vagus nerve, and the ability of the mucosal protection of the gut to fight off infection. And in fact, just as a note, we have been doing a lot of tracing studies in the central nervous system over the years, and almost invariably we see that there is a connection, a very specific connection between the central amygdala and the parasympathetic neurons of the vagus nerve. So there seems to be some sort of preferential circuit via which the brain can influence the parasympathetic system via the central amygdala. And in such a way that potential changes in the activity of the central amygdala will result in alterations in the normal functioning of internal organs, in particular the gastrointestinal tract, innervated by the gut, and in particular the Brunner's glands, in such a way that changes in this activity will cause alterations in the mucosal protection of the gastrointestinal tract. So one thing that we did, what this graph is showing is that when we record as many neurons as we could at the same time in the brains of behaving mice, what we observed that unlike in baseline activity, there was a very sudden and profound inhibition of the activity of the central amygdala as soon as we restrain the animals in using our stress protocol. So the activity of central amygdala seems to be very sensitive to restrain stress, at least in mice. And it's very different from what we see in normal activity, where these cells tend to respond very strongly when the animals are eating or interacting socially. So, to assess the possibility that the central amygdala is important for this system, we use the same chemogenetic technique, the same way to stimulate neurons by infusion of certain chemicals only in the central amygdala, and we observed whether this had any effect on peripheral immunity in the animals. So here, green again are the neurons in central amygdala expressing the receptor that we can activate, and these green dots correspond to neurons that the firing rates of which increased in response to the activation. So naturally, we see a lot of neurons activated in the central amygdala itself, but more interestingly, we see that whenever we activated the central amygdala, we see a potent activation of the parasympathetic neurons of the vagus nerve, meaning that this connection is functional. Here, this red signal here near the parasympathetic neurons are fibers coming from the central amygdala, because this projection from central amygdala to the medulla, where the parasympathetic neurons are, is very strong, as I mentioned before. So then we look at the effects of stimulating central amygdala in the parameters that we observed before. And again, we see that central amygdala activation is capable of restoring the normal counts of lactobacilli in the gut of stressed animals, just like the vagus nerve. again, we could no longer detect the presence of staphylococci, this pathogenic bacteria, in the blood of animals that had the central amygdala activated. So this is the levels of detection, just to remind you, in males and females. And also, just again, like the vagus nerve, we see a restoration of the normal size of the spleen, meaning that the body no longer needs to fight peripheral infection by recruiting cells that are stored in the spleen. Just as a side note, we found that this contraction of the spleen upon infection depends not on the parasympathetic system, but on sympathetic ganglia that innervate the spleen. But that's just a side note. What was important for us was to verify whether stimulating central amygdala was sufficient to drive the activity of the glands in the intestine that help lactobacilli to proliferate. So again, we implanted a stimulator in the central amygdala of mice. This is a stimulator that is very similar to what is used in techniques such as deep brain stimulation in certain patients. And we performed at the same time confocal imaging using calcium probes to observe the behavior of the glands. And as soon as we pulse the amygdala with electrical signals, we see again brighter cells, meaning that the calcium is being mobilized in the cells, which is an event that is a proxy for the mucus release of the cells. So again, this, sorry, I don't seem to be able to, yeah. Basically, that means that the central amygdala is a nucleus in the brain sensitive to stress that via the vagus nerve alters the activity of the glands. So what we think is happening here is that whenever we have alterations in central amygdala activity, this alteration will have as an effect a diminished tone in the parasympathetic neurons in the medulla. So if these neurons stop firing, these neurons will also stop firing. And that's why maybe central amygdala neurons respond so strongly during digestion and eating. And in turn, this diminished activity of vagal parasympathetic neurons will cause a change in the normal mucosal microbial environment in the gut, causing, in this case, a decrease of the amounts of mucin, of this gel released by Brunner's glands in the surface of the gut, which then prevents lactobacilli to proliferate in the gut. This will decrease the number of metabolites from lactobacilli that help to tighten the gut epithelium, allowing for harmful substances to cross into the blood. So this is basically a cartoon just rephrasing what I just said. So in states of rest and digest, which is the classical situation where the vagus nerve is more active, we have potentially more activity in central amygdala, more activity in the vagus nerve, greater ability of these Brunner's glands to release mucus, an ability of lactobacilli to proliferate and increase the expression of tight junctions, thereby increasing immunity. Okay. So I would like to thank the great people who have been working on this type of body-brain systems over the years. Chang Hao was the one driving all the bacterial studies for for this Brunner's gland project, and Wenfei Han, who is here, he was one of the first to really develop the intravital imaging system a couple of years ago in different projects with Leonardo, and also Tong, who has helped us with Peter Neckel in getting data from Brunner's glands in humans, including data that I was not able to show today, showing that removing this particular section of the duodenum in humans has immune implications that are not replicated by removing sections just adjoining this part of the intestine. And with that, I thank you for listening.
Well, thank you for this fantastic talk.
Thank you. Thank you.
I have been collecting a couple of questions, and one very interesting question is, what may be the evolutionary advantage leading to the leaky gut responses to stress?
Yeah, this is the question like everyone is interested in. It's a puzzling question. I totally agree. It's a great question, because why organisms would react in this way, I don't think anybody has a good explanation thus far. One possibility that has been raised is that these situations of chronic stress usually happen when animals are not being able to find food in nature. So the leaky gut could be a mechanism by which you maximize absorption of whatever nutrient you may find around you. Another explanation is that the body is saving resources for the immune system and the sympathetic system during confronting danger. But none of those are easy to test, and I don't know of any real mechanistic explanation for it, just the observation that this happens in almost every organism studied.
Very interesting. Another question was, are there other potential ways to stimulate the amygdala to impact the vagal nerve activity, potentially to be used in humans?
Yeah, so there are centers in different places that use manipulations of the amygdala in patients suffering from depression or severe anxiety. The problem is that the central amygdala is a very small nucleus really embedded deep into the temporal lobe. So to target with high specificity in humans is very difficult. One, let us say, physiological mechanism by which the central amygdala is activated is by eating, especially good food, like the neurons in central amygdala, especially this part that projects to the brainstem, responds very strongly to sweet taste and other palatable and other elements that could be called pleasurable, like social, sexual interactions. But for treatment, yeah, there is this limitation. Usually people say, I am in the amygdala, which is a problem because there are many nuclei there that are not really related to the central amygdala. Yeah.
Okay. There's also a question about gut-gastric operations, whether this could have also an impact on
gastric operations, yeah. So there was, we actually studied the potential effects of different ways to perform bariatric surgery in animals. The problem is there was a lot of stuff we couldn't fit it into the paper. But basically, we had two models that Wenfei developed in which in one of them, the glands are preserved, and in the other is not. So the route via which the food crosses the GI tract may or may not encounter the glands. And there was a difference in the way the organism responded to certain stimuli, especially infections, and the amount of mucus in the gut also changed. What complicates this is the fact that bariatric surgery changes a lot the levels of circulating hormones that are released by the gut. Normally they increase a lot. So these hormones I didn't show, but they also influence via the vagus nerve the activity of the glands. Okay. So the animals that have the glands preserved, they in some way look better than the normal animals because their hormone levels are very high. So it's something that needs some further thought until we fully understand what is happening.
Absolutely, probably very important for the patients. One of the many questions target the question of the timeline. How long does the stress have to be to induce such a change? Is it reversible? If so, how long would it take?
Yeah, that we didn't study the timing very carefully because we had to examine the internal organs of the animals. So we were very interested in the immune response, the bacterial response, the spleen, etc. So, but basically it's very clear that the stronger effects are produced by a more repeated treatment, like for example 1 week or 10 days was more or less in the range where we found it very hard to detect lactobacilli in culture. There is a type of bacterial culture medium called MRS that only in which only lactobacilli will grow and proliferate. So we use this as a marker for in addition to sequencing. And in this case, in this type of assay, this repeated stress for say 7 to 10 days dramatically diminish the number of bacterial counts in the animals. Yeah.
Okay, and there is an entire procedure that and the mechanism behind it, but one easy way of healing it would be just giving them more lactobacillus.
Well, yeah, this is also a good question. So, as we see in our study, lactobacilli and other microbes are very important for health, but they cannot exist or survive in the gut by themselves. So they need a whole environment in the gut that promotes their survival and proliferation. One critical element seems to be, among other things, the amount of mucus or mucins that are present in the gut that certain species, including lactobacilli, need to metabolize. These glands, they release a very specific type of mucin called mucin 6. And it could be that this type of mucin, its presence is very important. So I also didn't show the data, but if we take these animals without the glands and we give them just mucin 6, that not only improved their general state, but also we saw increases in the number of lactobacilli in these animals. So just the microbes alone in isolation may not be sufficient. You need a whole system, including favorable psychological states, to preserve the ability of this bacteria to proliferate.
Yeah, definitely. I think that goes hand in hand with the findings that microbiome environment has to be sustained, so to speak.
Exactly.
Mm-hmm. Okay. I think the number of questions are so many. I've been trying to sort those out that are mostly converging. But I think most of the questions have been answered. And I thank you so much for this wonderful talk.
Thank you for that.